Okay, welcome everybody to the second day of the Oxford Autumn School in Neuroscience. I hope many of you were able to be here yesterday. Today we've got a very different sets of symposia. And the first one is really circuit level neurophysiology. And the second one in the afternoon is about the effects of COVID on the mind and brain. So I hope both of those things will be of interest to you. So to begin with, we've got a symposium on finding a piece of the action, and that's really code for let's work out how physiology affects behavior. And it's being chaired by my colleague Andy Sharot, who is a program leader at the MRC Brain Network Dynamic Unit here in Oxford. And Andy's leading a team there, which is looking particularly at the neurophysiology of the basal ganglia, and he'll be giving a talk on that later on. But perhaps I should ask Andy to introduce the symposium and tell us what the context and content will be. Thanks, Andy.
So good morning, everybody. So as Masood said, the first session today is going to be all about movement, and particularly the neurophysiology of movement. So to some degree, one could argue that deciding when and how to move and how to do it better is really a fundamental, or even the fundamental function of brains and nervous systems. And so the speakers today have been selected to give different windows on mechanisms, specific circuits in the brain, which carry out specific parts of controlling movement and action, and also how different factors like sensation affect the way we move. So without further ado, I'm going to hand over to our first speaker, Ian Duguid, who is professor at the University of Edinburgh and also a senior Wellcome Fellow. So, Ian.
Okay, well, thank you, Andy, for the introduction and the opportunity to come and talk to you about a project that we've recently completed that looked at how different brain areas interact. So how different brain areas interact to both initiate and control voluntary or purposive movement. So what do I mean by that? So as mammals, and especially humans, we can execute a wide variety or a wide repertoire of motor behaviors or motor movements. So here's an example here, where it's a top-down view of a human who's controlling a manipulandum or a lever. And by exerting force on the lever, it can move that lever around. And this particular subject is waiting for an external cue. That could be an auditory cue, or it could be a visual cue. And upon presentation of the cue, we'll just move the lever, exert some force, move the lever from the start position to the target position. And this is a cue or a sensory triggered purposive or voluntary movement. And humans can do this, but equally, what I'm going to focus a little bit more on is our sort of model of choice, which is the rodent. And rodents also can perform these types of behavior. So in the wild or in the laboratory, a rodent can be trained to wait for a sensory stimulus. So again, that could be an auditory stimulus or visual stimulus. Upon hearing the stimulus, it will then execute a motor movement or a goal-directed behavior to then receive some form of water reward, or in this case, a water reward. So if we break that behavior down into its component parts, you have a sort of resting phase where the animal is waiting, and then there will be some form of sensory cue. When the cue is presented, the animal will then, or the brain will then process that information, and then at some point distant to that, the animal will start to initiate a movement in response to that cue, which is the motor output. But the time delay between the cue and the initiation of movement is referred to as the reaction time. It'll execute the movement, and then it will receive some form of water reward. And what we're really interested in is this time point here, the initiation. What brain areas actually contribute to the engagement of the musculature to actually engage in the motor behavior? So what brain areas actually drive this movement of initiation, or the initiation of purposive voluntary movement? So if we take a step back again, this is looking at a sort of slice through a rodent brain, and it's a simplified version of the schematic representation of the different areas of the brain that might be involved in controlling voluntary movement. So we have the motor cortex, which is up here. So that's the area which generates patterns of activity or patterns of motor commands that descend down into the brainstem and into the spinal cord, that engages the skeletal musculature and then executes a particular behavior. You also get feedback from the periphery that comes into the brainstem, goes up into the cerebellum, but you'll see that this direct sort of commands that are coming down also go to the brainstem and back into the cerebellum. The cerebellum is thought to receive information about the intended motor command, and then it gets feedback from the periphery to say, okay, if I intended to move my limb, what was the intended goal? And then I get feedback from the periphery to say, okay, but where is your limb? And how close are those two commands? If there's a difference between that, the cerebellum processes that and sends that signal back through thalamus, back to the motor cortex to update those patterns of motor commands. That's sent down to the brainstem and the spinal cord to adjust and adapt that ongoing movement. And you can see that there's clearly distinct motor loops where there's information going around these loops the entire time as the animal is engaged in a behavior. So historically, what led us to be interested in what brain areas are involved in the initiation of movement. Here's some work done by Thatch back in 1975, where they're using primates who were, again, trained to do a volitional forelimb movement. And they recorded from the cerebellum, which is up here. So this is basically just a histogram of activity. So just to think of this as changing activity over time, where they're recording from individual neurons within the cerebellum. And the important point is this, this arrow shown here, which is the onset or the initiation of the movement. And it's quite clear to see that in terms of activity, there's activity that precedes or is left of the initiation of movement. So the cerebellum seems to be active before the animal actually engages in the movement. Whereas if you look at the bottom down here, this is activity that was recorded in the motor cortex. So as I mentioned, that's the area that actually sends the motor commands that go down to the spinal cord to engage the movement. And that the activity in motor cortex is in or around movement initiation, as you'd expect if it's sending the signals to actually generate movement. So it's interesting that in the cerebellum, there seems to be this activity that precedes movement. What if we go to the next node along? So cerebellum, thalamus, motor cortex. Do the same sort of recording from units in motor thalamus. And this is a primate that's doing a center out reaching task. So any of these targets are illuminated and the primate then reaches towards that target. So we see the target stimulus coming on here. And then at some point distant, there's a reaction time where the animal then reaches towards a target. And this is the movement of the arm. And then once it reaches the target, it obviously gets a water reward. But if we look at the activity again, recording from thalamus, and this is what's shown here. This is the activity of the neurons within thalamus, or a particular neuron within thalamus. This is the start of the movement, and this is the end of the movement. But again, you can see that activity actually rises before the onset of the actual movement, before there's any detectable movement of the forelimb. So both in cerebellum and in thalamus, there seems to be activity that precedes the onset of the directed, goal-directed movement. So when we go back to our schematic diagram, the question, given this sort of the historical evidence to suggest that, okay, the cerebellum is active preceding movement, the thalamus is active preceding movement. So to what extent does the cerebellum actually play a role in providing the information that's necessary to execute the movement? And does this information actually go from cerebellum through thalamus back to motor cortex? So how do we go about interrogating the underlying neural circuitry? So to do that, I said our model of choice is the rodent, and in this case it's mice. So we developed a cued linear forelimb push task for mice. What does that look like? So on the left-hand side, you can see the animal is resting on a movable lever on its left forepaw and a static lever on the right. There's a cue that's presented. It's a 6 kilohertz auditory cue. When the animal hears the cue, it's trained to then push the lever forward about 4 or 5 millimeters. And if it pushes the lever forward to the forward position, it receives a 5 microliter water reward. Okay, so that's a successful hit trial. But equally, the animal or the mouse can hear the cue, but chooses to ignore it. This is classed as a mistrial. There's no reward. And then this goes back to what's an intertrial interval of between 4 and 6 seconds, basically a waiting period before another cue is presented, and then the animal can choose whether to push. Sure. And so there's an intertrial interval, and then the animal can engage in a subsequent trial. So what does that look like? Here's a video. Here's a mistrial. So the animal here's the cue. That's where the green spot is presented. Doesn't push. The second one's a hit trial. Here comes the auditory cue. Pushes the lever forward and then receives a water reward. So mice will learn to do this repeatedly in a 30 to 60 minute training session. They'll do this many, many times. It's a relatively simple behavior, but it has all the key components that we look for. There's a quiet period. There's a cue or a sensory stimulus. And then there's a reaction time, and the animal then engages in the desired behavior. So they learn this over 2 weeks. They learn to do it reproducibly and do many trials. Over time, the reaction time comes down. So the time between the cue and the actual engagement with the lever comes down. They're quite sloppy for a start, they're 2 to 3 seconds, but when they become expert at it, within 300 milliseconds after the tone, the animal is engaged in pushing the lever. So they're keen to get the water reward. As I say, they'll do about 100, 150 trials within a behavioral session, but they're not faultless. You know, it's a long session for a mouse to sit there. And you can see down here that they engage about 60, 70% of the time, because at some point they'll become sated, they might disengage with the task, but you still get about a 70% success rate. So we have a highly reproducible trial-to-trial behavior that then we can try and look and interrogate the underlying neural mechanisms that would be driving this initiation of movement. Okay, so the thalamocortical, cerebello-thalamocortical pathway, that's the 3 key nodes that we're interested in. The cerebellum, projecting to the thalamus, projecting to motor cortex. So is there this pathway present, and to what extent is this a major pathway? So what we did here is we basically just used sort of modern day anatomy tracing techniques to map that projections from cerebellum, which is over here. And you'll see these different terminology. The DN-IPN is basically just 2 of the output, it's dentate and interpositus nucleus, but they're 2 of the output nuclei of the cerebellum. So and these project into motor thalamus, which you'll see here, MTH, and this projects up to a region of motor cortex, which is the caudal forelimb area. So it's the area that's actually controlling the forelimb. Well, this is sort of the cerebellum through to thalamus up to motor cortex. And what we use is 2 different tracers. So one is a retrograde tracer. So this will identify cells which are projecting from thalamus to motor cortex. And then we inject an anterograde tracer, which goes from cerebellum to thalamus. And we're looking in thalamus for the region that projects to motor cortex, but also receives input from the cerebellum. And that's what we see here. So this is an area for the aficionados. This is an area of motor thalamus, which is called the ventral anterior lateral motor thalamus. But it's a region that receives or projects to motor cortex, which is seen here, and receives an abundant number of axons from the cerebellum, which is seen here. And if we look across animals, and take a sort of average density plot, there's clearly an area that receives strong cerebellar input to then project that information to motor cortex. And in terms of how strong or how the degree of connectivity that's between cerebellum and thalamus, it's extremely high. We're surprised to find that across a number of animals, when we basically use an antibody that stains for the axon terminals that would come in from cerebellum, and see to what extent they're in close apposition or synapsing onto cells which project from thalamus to motor cortex. And what we found was that we were struggling to actually find any cells in that area of motor thalamus that didn't receive input from cerebellum. So a really high degree of connectivity, a really strong input that's coming from cerebellum through this area of thalamus up to motor cortex. So we're interested in whether these brain regions are actually concerned with the initiation of movement. Are they providing a signal that would actually trigger the movement? So how can we go about trying to disentangle the activity along that pathway? What we did initially was to opt for a recording configuration, which is imaging we were using. So this is using a calcium indicator, which is a proxy for activity within cells. So we're using this reporter GCAMP6. We inject that into cells within motor thalamus, and then we use a relay lens, which picks up the photons when calcium binds, GCAMP fluoresces, picks up the photons, and these photons can then be imaged using an objective and a 2-photon microscope. So this gives a way of imaging activity deep within the brain. And when we do that, so on the right-hand side is a video of, this is a field of view that we would see through our grin lens. And then I'll run that, it's 4 times speeded up, but the cue will be presented. You see there's a relatively short latency rise in activity that occurs across a number of neurons within thalamus. And this can be seen reproducibly, trial on trial. So this gives a sort of way to assay a large number of cells within motor thalamus while the animal is engaged in our behavior. So here's a representation of a large population representation of the activity that we see. So if you just think of each of these rows across here as a single neuron, and this is a representation or a pseudo-color representation of the change in the fluorescence of that individual cell as in a proxy for its activity. So here it goes from baseline, which is relatively white, and then rises up to a hot color when the fluorescence rises and the activity of that neuron increases. So I think it's quite clear to see that across all of the neurons that we recorded from, there are a large number of neurons that when we've identified here, which is in the purple spots, are the onset of that activity change, when it starts to rise. So you can see there's a large amount of activity that actually precedes MI, which is movement initiation. So a lot of activity that's occurring within thalamus precedes the onset of the movement, which is relatively similar to what we saw in the primates earlier on, or the studies that I showed you previously. We also see cells that decrease their activity. So they're reducing their activity, again, preceding movement initiation, which is this dotted line. And there are cells which show activity that happens after the movement has been completed. But if we're interested in cells that can contribute to the movement, then we're interested in these cells that actually are active prior to the onset or the initiation. So, because these can actually contribute to the movement. So if we look at these cells, the vast majority of these, about 80% of cells show an increase in activity, and about 20% show a decrease in activity. And it doesn't really matter in terms of where you look. This is just a spatial representation looking down on top of motor thalamus. And they're color coded, so these dark green cells are the cells that are increasing activity. They're pretty much peppered across the entire thalamus, so there's no topographical organization. But to say that the predominant response in motor thalamus is this early onset increase in activity. Okay, but what if we then have a look at differences in the actual behavior? So here's a representative field of view that we've been looking at in thalamus. Color-coded in green are these cells that show early onset activity. And the behavior of the mouse is not stereotyped, it's not robotic. So in some trials, mice will have a very fast reaction time, 100 milliseconds. In some trials, it might have a moderate reaction time, which is 200, 300, 400 milliseconds. And in some, it's quite sloppy, and it can be 600, 700 milliseconds before it actually engages with lever. So we can split these reaction times down into short, medium, and long, and that's the color coding here. And then we're just selecting an individual cell and looking at the change in fluorescence, so the change in activity of this cell, with respect to the reaction time. So here is a long reaction time. So that's when the cue is presented. The fluorescence of this particular cell stays pretty flat. And then just before movement, about 200, 300 milliseconds before, you see this rapid rise in activity. So that cell seems to have a fixed latency of activity change, irrespective of where the cue is presented. So here's a long reaction time, a medium reaction time, and here's a short reaction time. It's flat until that 200 to 300 milliseconds before. Okay, that's one cell, but what if we look at all of this population together in the field of view, sum their activity, and then present it in the same way? But again, long reaction time, short, medium, and short reaction time, fluorescence is flat until about that 200, 300 milliseconds before, and then there's a rapid rise in activity. So it seems to suggest that these thalamic neurons that are active before movement have a fixed latency timing signal. Okay, so that would be fitting in terms of if you need a signal that goes to motor cortex to actually initiate a movement, then you want it to really be fixed to actually trigger that movement exactly when you want to move. So I've introduced the three different nodes along this pathway. We have the cerebellum, which is over here on the right. We have the thalamus, and then we have motor cortex. So the activity seems to correlate with having a role in movement initiation, but actually, are they necessary? So what we did was we injected a very small volume of muscimol, which is a drug that inhibits activity within a particular region. So when we inject that drug, it'll silence any activity within the region. And when we do that, when we inject it into the cerebellum or the cerebellar nuclei, across here is basically the number of successful trials. And in green, when we inject or inhibit the cerebellar output, you get about 90% reduction in the ability of the animal to actually engage with the lever and generate the lever push or the task. If we inject saline, no effect, which is what you'd expect. It's just a salt solution. It shouldn't have any effect on the behavior. Okay, so that suggests that definitely output from the cerebellar nuclei are essential for or necessary for initiating this lever push movement. What if we do the same in thalamus? So again, we're just injecting. This is actually showing you the diffusion of the drug within thalamus. We see pretty much the same effect, about a 90% reduction in the ability of the animal to engage. And again, saline has no effect. So clearly, both of these nodes along the pathway, their activity is necessary for the animal to execute the behavior. What does it look like when you inactivate? So this is a video of inactivating motor thalamus, so that central node. What does it look like? And the left-hand side will be control. And if you just look at the right-hand side, that's the muscimal inactivation of thalamus. So when I play the video, if you look on the right-hand side, the green dot will appear when the cue arises. You can see that the animal's whisking. It understands and it senses the fact that the cue is being presented, but it just can't initiate that learned movement. It can still move its forelimb, but it can't generate the learned movement that it's been trained to perform. Whereas in the control, it's perfectly fine. So again, that would suggest that there's this signal that's traveling through that pathway that's necessary for the execution or the initiation of that movement. Why is that? I'm not going to go into all the details as to why, but what we did was to record in the motor cortical region, so recording from the cells that would actually be outputting to the spinal cord that would generate the contraction of the muscles. And we inactivate thalamus. Before we inactivate thalamus, there's just a recording from a single unit or a single cell within motor cortex, and you can see it as normal. In this particular cell, there's a depolarization or an increased activity. When we inactivate this particular brain region, it completely suppresses that change in activity that you see in cortex. So by inactivating thalamus, you reduce the drive that would be going to motor cortex. There's no longer a drive, there's no longer a signal that's there. Motor cortex doesn't change its activity, and therefore the animal doesn't engage in the behavior. Okay, so that's we've shown evidence that if you inactivate the different nodes along that pathway, then it stops the animal initiate the behavior. But can we do the reverse? Can you actually stimulate those nodes or those cells within those different brain areas to actually generate the movement itself? So how did we go about doing that? So here's a schematic of the experiment that we did. So here we're injecting a channel rhodopsin, which is a light activated protein, so that under viral control. So we transduce neurons within motor thalamus with this light activated protein. So when we shine light on that area, depolarize the cells, it'll excite the cells, and then it'll send a signal propagating forward. And what we do is we put two light stimulation devices in place, one directly above the cells within thalamus, but then a second one that's placed in motor cortex itself. And what we're doing there is stimulating the axon terminals that would come from thalamus to motor cortex. So we have an element of specificity there that only this region will be activated, or the axons within this region will be activated. But when we stimulate, or when we enter the animal into the behavior, when you present the cue, it's what it's learned to do. And here's a quantification of the probability that the animal will push the lever. Not surprisingly, when there's a cue, it's what it's trained to do. 60 or 70% of the time, when it hears a cue, it'll push the lever forward. But when we remove the cue, so there's no auditory cue given anymore, and then we just stimulate either thalamus or its axon terminals in motor cortex, about 30 to 40% of trials, the animal will push the lever as you would see in a normal behavior. So that's just stimulation with no cue there. There's no external stimulus. You just stimulate that pathway and the animal then engages in the behavior. What does that look like? So this is direct stimulation of thalamus itself. And again, if you look at the right-hand side, that's when you'll see when the laser was switched on. When the laser switched on the right-hand side, you can see it pushes the lever. So there's no cue. You just stimulate that pathway and it pushes the lever. Very similar to what you would see when you present the cue. You can see that there's a difference in terms of the licking nature, because we train the animals that they don't get a reward when you photoactivate that pathway, because then it can extinguish the behavior that would be driven by the cue. Okay, so present the laser and you get a short latency push. And that's the same irrespective of whether you stimulate thalamus or its axon terminals in M1. Why is that? Because one of the arguments with optogenetics is it's a very blunt approach. You're just shining light. You know, I showed you before that the signatures in thalamus, some go up, some go down, and we stimulate all of these cells and push them all to increase their activity. But here's just a recording that we made from a single neuron in motor cortex. And here is just showing the change in activity. Black is when we present the cue, and in interleaved trials, when we then stimulate the pathway, is in green. And you can see that they're highly overlapping. And actually, if you look across a number of cells, there's quite a tight correlation that the change in activity you see when there's a go cue somewhat mimics the activity that you see when there's photostimulation. So maybe not surprisingly, when it gets the same signal into motor cortex, it executes the same behavior. Okay, so that's stimulating the thalamus. What if we then go one step back and ask, okay, what if we stimulate the output from the cerebellum? So again, we're expressing this light activated protein. It will also go into the axon, so we can stimulate the axon terminals of this input from the cerebellum in motor thalamus to see if we get the same effect. And here we're recording with a high density silicon probe in motor cortex to see what changes in activity we see in cortex. So again, not surprising, this is the probability that the mouse will engage and push the lever. Cue, it's what it's trained to do. So 60, 70% of the time, you see a lever press, you emit the cue, and then you just stimulate that pathway. Again, 30, 40% of the time, the animal will push the lever. What does that look like? Again, look at the right-hand side. So it's a direct stimulation of that cerebellar output. Present the laser, and you get a recapitulation of the learned behavior. And again, if we look across, so this is us recording from motor cortex. So we're 2 steps removed. So you're stimulating cerebellum, but recording from motor cortex. Here's just an example of recordings from single units. So each of these dots in the raster is when the neuron spikes. When you present cue, you see that there's an increase in activity around movement initiation, which is what you would expect. It actually happens before. And this is the same across all 3 units. If you look at the Brack traces, and this is just a recording of the spikes or the activity of that cell over time. Then we stimulate, and similar to what we saw before, you get a pretty good recapitulation of the activity pattern the motor cortex normally sees when you stimulate the cerebellar input. And again, if you look at the correlation across here between activity in motor cortex in Go trials versus activity when you photostimulate with no auditory cue, it's a very, very good correlation. So we're really basically mimicking the Go cue by stimulating that pathway. Okay, so just to finish off in the last 2 to 3 minutes, if we're just stimulating thalamus or this pathway, and it's just activating the muscles, it could be the fact that the animal is just sitting on a lever, so it can do nothing else but push the lever. So it's not really a recapitulation of the behavior. So is there any element of behavioral context? What do I mean by that? Well, this is the learned behavioral context. The animal sits in a tube, has 2 levers. The left-hand side is movable. There's a cue, that auditory cue that we talked about. There's a lever and obviously gets a water reward when it successfully executes the trial. But then we place them into an altered behavioral context. So it's still head restrained, but there is still a cue. There's an auditory Go cue, but there's no lever and there's no reward. So the animal is just sitting on a flat surface. So if we stimulate, what do we see? So in the learned behavioral context, this is now a tracking of the forelimb. So it's a sort of vector trajectory of the forelimb. As you're expecting cue, there's forward motion, backwards, up and down. With the cue, the animal just pushes a lever, and that's basically the trajectory. The tracking of the limit is the trajectory of the lever movement. And in that 30–40% of trials, when you stimulate with no Go cue and the animal pushes a lever, not surprisingly, you get the same vector trajectory. But if we transfer the mice to an altered behavioral context when there's no lever, when we present the cue, the animals have learned there is no lever, there is no reward, I don't need to move. And that's what we see here. There's no black vector trajectory. It's just basically sitting there. But when we stimulate, we don't see a push-like behavior. We almost see the opposite. It's actually a retraction and the animal moves its limb up and off the ground. So this is suggesting, well, actually the stimulation doesn't just co-contract muscles and you get a push-like, irrespective of where the animal is. There's an element of behavioral context dependence. When you stimulate that pathway, it'll execute movement. And that's what's shown here. If you look at the left-hand side, the right-hand side, probability of any movement is similar whether you play the cue in the learned behavioral context or you stimulate that pathway. But even if you take them out of that behavioral context, they will always move. But whether they generate push-like movements, they never do in the altered behavioral context. It's only in the learned behavioral context that they execute that push-like behavior. So it's not just that we stimulate pathway and you'll get the same behavior each time. It's a learned behavior depending on which context the animal is placed in. And again, if we open them up into, so this is non-head restrained now. This is locomoting around in a open field. And when again we stimulate, this is the probability of movement. We see discrete movements because that's what you'd expect. It always moves when you stimulate that pathway. But we don't see push-like movements. We don't see initiation of locomotion or grooming, which are rhythmic behaviors. So it's very specific in terms of what movement is generated, depending on which context the animal is in within that time. Okay, so just to summarize and finish up, then I hope I've given you some insight into the fact that this cerebellar thalamocortical pathway seems to convey a reliable timing or feed-forward timing signal that's necessary for movement initiation. If you silence the pathway or any of the nodes along that, it blocks movement initiation. And if you do the opposite and you stimulate any node along that pathway, then you can generate or trigger movement even without an external sensory stimulus. And the last part that I showed you was that movement or the movement that is generated is entirely behavioral context dependent. So that just leads me to acknowledge the people who did this. Josh Dacre did the lion's share of this work, and the rest of the people contributed within the lab. Also had help from colleagues at Ganglia Farm Research Campus, Adam Hantman and Natalie Rochford in Edinburgh, Benjamin Groovy, Young Rundabin, Thiago Branco, the Wellcome Trust, who have and continue to fund most of the work that we do, and the rest of our funders. And happy to take questions.
Thanks a lot, Ian. It's really beautifully clean data, very convincing. So do we have any questions from the room? Raphael.
Thank you for a fascinating talk. So it seems that I'm asking you, train behavioral complex have this representation almost ready for you to activate. So in these neurons which were increasing active, do you see that their baseline firing before we provided a stimulation was high? So do you see this kind of representation being ready?
In motor cortex? That is something we're actively seeking now as a follow-on question. We haven't got an answer as yet as to whether you mean that they're predisposed to be. Yes is going to be the answer. There's definitely almost, I guess the description would be almost a pattern completion where there's information coming in. And when I said when you inactivate thalamus that it suppresses the activity, that's not the whole story. It suppresses about 75 to 80% of the activity. There's still a residual 20%. And that's not surprising because you're bound to have contextual information coming from the brain, sensory information about its environment, whether it wants to engage in the task at that time. So all this information is definitely there. And that's what we're trying to unpick now. What is driving that? Is it there constantly? You might say if it's context dependent, exactly as you say, it's there the whole time and it elevates the activity or it changes the dynamics. Or is that information timed? Again, it comes in from the cue, provides additional information, and then the timing signal is what kicks that over threshold and then has a sufficient level of activity to drive the movement. We don't know the answer yet, but it's obviously a super interesting question to go after.
One or two more? Yeah.
Yeah, so I saw that there's something very unique in that the activation difference in activation. So I'm also asking that I successfully strong activation of the challenge.
Yeah, so we looked into that and we didn't. It's hard because there's a motivational aspect to it because we also have different behaviors within the lab that we give a 250 millisecond auditory tone. This is a 2-second tone, okay? And it's almost the mice work out what is the energy efficient way to do it. And when you give them a 2-second tone, they have a wide variability in reaction times. If you give them a 250 millisecond tone, they have incredibly tight and accurate reaction times. So I think there's an element of how it's engaging with the task. And yeah, it would be input from other parts of the brain that we're not yet sure of where that's coming from that would then give this variability. But we couldn't find any tight correlation with, but the signal follows. So if it's a fast reaction time, the signal doesn't change. If it's a long reaction time or short reaction time, that signal in terms of the thalamic signal doesn't change. It's always 200 to 300 milliseconds before. So it's almost irrespective of cue. It acknowledges the cue, but that pathway is not concerned with integrating information over time. It's just almost waiting, and then there must be some other brain regions that are triggering that 300 milliseconds before, send the signal because now is the time at which I want to move. So there's a lot more depth to this, and this is just a signal that's coming through. But as I showed in the circuit diagrams, there's a lot of loops, sensory motor loops. So trying to unpick, and how to unpick, we've shown a part of a loop, but by definition, it's a loop. So we want to then follow that information as it goes around the loop to see, well, is it this bit that's pivotal, or is actually it just a sort of a starting point to then drive activity around the entire motor loop?
Yeah, I think in the interest of time, we better move on, I'm afraid.
Okay.
Thanks again, Ian. Do I put the video on as well?
Yes.
Okay, so it's my pleasure to introduce our next speaker, Anne Duan. So Anne did her PhD with Carlos Brody in Princeton before moving on to a postdoc in Shanghai, and she's just started her lab at the Sainsbury Wellcome Center at UCL. And she's going to tell us about the importance of Calicular circuits in flexible action control. So over to you, Anne.
Thanks, Andy. Can you guys hear me okay? Okay, so thank you for the invitation and thanks for the kind introduction. It's my pleasure to be part of the Oxford Autumn School this year and hope everyone's having fun so far. So today I'll talk about my work on cortical calicular circuits for flexible control of action. Normally my talk for the same material is titled Cortical Calicular Circuits for Decision Making. But the theme of our session this morning kind of inspired me to think about what is the goal of decision making most of the time, which is, if you think about it at its core, flexible control of action in response to environmental changes or internal state modulation. And to understand these processes, we need to think about how information maintained in the brain in absence of sensory stimuli or motor action is used to guide complex behavior. And although a lot of these cognitive internal states have mostly been associated with areas such as frontal cortex or parietal cortex, what we've come to realize is that these internal states are actually represented in the brain in a distributed and redundant fashion in a densely interconnected cortical and subcortical network. And some of these subcortical areas that have not classically been implicated in higher cognition may actually play a similarly important role as cortex. And today I'll be focusing on a key node that is missing from Ian's diagram, the midbrain superior caliculus. Okay. So the mammalian superior caliculus, or optic tectum in other vertebrates, is an evolutionarily conserved brain structure, sensor motor structure that consists of distinct cell layers, which can be functionally divided into the superficial and the deep layers. So the superficial layers receive direct retinal input, and they're important for visual processing. The superficial and the intermediate and deep layers receive multisensory and modulatory input, and they send descending projections to motor nuclei. So if you think about it, actually, the deep layers of the SC are interconnected with virtually the entire brain. So it's well positioned to serve as a functional hub that combines multisensory and contextual information to guide behaviors. However, despite a rich literature in SC's sensory and motor functions, and more recently, its role in target selection and spatial attention, SC's importance for flexible control of action or higher cognition in general remain much less investigated. And that is what I will be talking about today. So I feel real thankful that Ian has given a lot of background as well as explaining the method. So hopefully that'll save me some time, and I'll do the crazy thing of explaining 2 stories in a 30-minute talk. We'll see how it goes. I'll start with a series of work on calicular circuits for executive control in 3D moving rats, and then move on to work using half-fixed mice to study motor planning. So essentially the first story is about flexible sensor motor routing, and the second story is about how information is maintained before delayed action. Okay, executive control. Let's first define what it is. Here I'm referring to the ability to flexibly switch sensor motor routing to match the appropriate environmental context. For example, most of the time when you hear a phone ring, the automatic response is to pick it up and answer. But if you add a sound like this one, you might quickly form the response of silencing your phone when you hear a ring. But as soon as you step out of this auditorium or Zoom meeting, you can instantly change your response to the same stimulus based on the new context. This kind of stimulus response routing can be flexible and extremely rapid, and it can be studied in the laboratory using behavior paradigms such as the Stroop task, where subjects are instructed to either read the text of each word or name the color of each word. For example, this stimulus corresponds to orange in the word reading task, but to blue in the color naming task. Subjects can switch between these tasks on a very fast time scale, and they display interesting asymmetries where word reading is usually quick, easy, and prepotent, and the color naming is slower and harder. So to study the underlying neuromechanisms in rodents, we set out to develop an analogous behavior in the rat. We were inspired by the primate per-antipsychotic paradigm. So we developed a rapid task switching behavior where rats experience two types of trials, pro or anti. The trial type is indicated by a sound at the beginning of each trial. A silent delay will follow, during which rats have to remember the trial type. And then after the delay, a target stimulus will be presented as either the left or the right light. If it's a pro trial, rats should orient towards the light for reward. If it's an anti-trial, rats should orient away from the light for reward. Rats can perform fast rerouting between pro and anti-trials, either across blocks or in a fully interleaved version of the task. And it's important to note that consistent with primate paradigms, rats show multiple behavioral asymmetries between pro and anti trials, where pro is faster and easier to learn and execute, and anti is more cognitively demanding. So these asymmetries establish the rat pro-anti behavior as a comparable model to study flexible sensory motor routing and response inhibition. So how does the brain do it? We tested two brain regions that have been implicated in the pro and anti pathway respectively, the superior Caliculus and the medial prefrontal cortex. The SC receives direct sensory input, and the corresponding motor projections are essential for orienting to the contralateral side, motivating the idea that SC mediates the stimulus-driven pro responses. On the other hand, PFC neurons are hypothesized to encode the relevant task goal and allows anti-performance by inhibiting the pro pathway. So to test their causal roles, we conduct the reversible inactivation of SC or PFC, also using mucimol, and we expected the two inactivation to have the opposite effects on behavior. But instead, we were surprised to find that both SC and PFC were required for the anti-task, so they have sort of a similar role instead of the opposite roles. So if you're interested in the details of this, you can check out our paper. But we were more curious about, so these data argue against the idea where SC needs to be inhibited for anti-performance, and instead suggest that SC plays an active role in anti-task. But we then want to ask, what specific role is that? To ask what circuit mechanisms underlie SC's specific role, we conducted eFIS recording and optogenetic silencing of SC neurons and computational modeling of SC circuits. So I'm going to jump into some single neurons. The single neurons in SC have very heterogeneous task information. So shown here are the pro-anti task selectivity in well-isolated single neurons. So each row is a neuron, and the color represents pro-anti selectivity for that neuron as a function of time. And the neurons are sorted based on the timing of their peak selectivity. So you can see that there's no apparent order in terms of whether more neurons prefer pro or anti in any behavioral epoch. And if we take the same order of neurons and look at their choice signals, it's also completely mixed regarding their preference for either the ipsilateral choice or the contralateral choice. And it doesn't seem like there's any systematic relationship between task and choice selectivity among all the units. So the signals are there, task relevant signals, but it's hard for us to say much regarding the function of this region because individual neurons are highly heterogeneous. But it does look like there are two distinct subpopulations of SC neurons. One group that differentiated between pro and anti trials most strongly during the auditory cue, and a separate group that maintained task selectivity after the cue was no longer present, and we call them the delay choice neurons. To evaluate the amount of task context information in these SC subpopulations, we used a cross-validated linear decoding approach to classify pro versus anti responses in the population neural space. So here 1 means perfect classification, 0.5 means chance level performance of population decoding, and the vertical lines separate the cue, the delay, and the response epochs. So we found that the information of task context by SCQ neurons was as predicted, progressively weakened after the end of the cue. But interestingly, performance did not differentiate between correct and error trials. So this is consistent with a purely sensory signal. In contrast, the task context representation in the delay choice neurons was entirely disrupted on error trials, indicating a strong correlation with behavior. So we also look at the population decoding for choice. The SC delayed choice neurons, again, I should emphasize that they're defined based on the timing of their per anti-selectivity. It has nothing to do with their choice activity. But we found that these neurons have a very early representation of the correct choice, substantially faster than the SCQ neurons or different prefrontal cortical population. Here just shown one, but we actually recorded from multiple regions in the prefrontal cortex, and they're all slower than the SC delayed choice neurons. So these data strongly suggest that maybe the SC delayed choice neurons might be computing sensory-motor routing. So we actually focus on these neurons for further analyses. And we also zoomed into the moment immediately after the target stimulus was presented, because this is when animals can combine per anti information with sight light information to compute the correct choice. What we found is that most contra-preferring delayed choice neurons tend to be pro-preferring, and the IPSI-preferring delayed choice neurons tend to be anti-preferring. In other words, in contrast to the heterogeneity initially observed in the entire SC population, delayed choice neuron during this critical epoch had a systematic relationship between each neuron's per anti-selectivity and choice selectivity. We refer to these neurons at the ends of the spectrum as the pro-contra preferring and anti-IPSI preferring neurons, and they'll form the basic structure of our SC models later on. First, we want to have some data to constrain the models. We tested the requirement of SC activity during distinct task epochs via optogenic silencing of bilateral SC neurons during the Q period, the delay period, and the choice epoch. So Q period inactivation did not lead to a behavior deficit, consistent again with the sensory role of these signals. In contrast, delay period inactivation significantly increased error rates on anti-trials, suggesting that an intact representation of the task context is required in the SC during the delay period for correct task performance. And finally, given the early and strong choice signals in the SC, we were really surprised to find that bilateral choice period inactivation did not have an effect on accuracy, suggesting that SC dynamics during this epoch might be substantially more robust to perturbation than during the delay epoch. We want to get a more formal interpretation of these results, so that's when we turn to modeling. And the goals of the modeling exercise are twofold. First, we want to ask if there exists any set of parameters of Calicular models that could fit our optogenetic data, which turned out actually to be non-trivial. And second, if such circuits exist, we want to ask, what are the features that these circuits require for them to fit our data? We used a simplified SC model inspired by our EFIS data, where the circuit consists of 4 pools of neurons, a pro-anti-pool and a pro-contra pool, and an anti-pool on each hemisphere of the model. The pro units get activation on pro-trials, anti-units get activation on anti-trials, and motivated by past literature, we took the pro-contra neurons as the ones that are driving the motor choice. So the final choice of the model is determined by which of the 2 pro-contra units have greater activity. And we have other parameters of the model that include the sign and strength of connections, the degree of optogenic silencing, and so on. So can such an SC circuit replicate our experimental data? Large-scale computational searches for solutions reveal that the answer is yes. We found model solutions that can fit the delay and choice period activation data, and the dynamics of these solutions parallel those found in our EFACE data. I should say that although finding such a solution was not that easy, but considering the powerful computational searches we did, the result of we have a model that fits the data is not that satisfying or even that surprising. So we then want to ask, what are the properties of model circuits that could fit the data? To do so, we produce not only one solution, but many solutions. So here's a low dimensional representation of model dynamics from 300-something solutions. So each solution corresponds to 1 point in this plot. And you can see that the dynamics across solutions were highly heterogeneous. But despite the highly varied dynamics, as well as parameter values in different solutions, we identified 2 features that were consistent across model solutions. The vertical weights from anti to the pro units and the diagonal weights from anti to the pro units. Almost all solutions had inhibitory connections from the anti-unit to the pro unit on the same hemisphere, suggests that the suppression of the default pro pathway by anti-task context representation locally within the SC is important for response inhibition. Also, most solutions had excitatory connections from anti-unit to the pro unit on the opposite hemisphere. This constitutes a pathway for vector inversion on anti-trials because, as you know, that's the pro unit that needs to win for an correct anti-trial to occur. So we identified features of SC circuits that correspond to key processes of executive control. First, to inhibit task inappropriate action, and second, to execute task appropriate action. To summarize this part of the talk, we found that SC neurons represent task context during the memory period, and this activity is necessary for flexible sensory motor routing. A functionally defined subset of SC neurons, the delayed choice neurons, likely lead choice computation. And dynamical SC models reveal key mechanism of executive control within Calicular circuits. What I didn't have time to talk about today is that even though frontal cortical neurons have slower choice signals, neurons in PFC also represent pro-anti information. They have choice information, and mucin activation of PFC also resulted in an impairment in behavior. So if we want to understand how PFC and SC cooperate during flexible control behavior, we need to examine PFC and SC together in a cell type and pathway specific manner. And that's when I turn to the mouse model, where the genetic tools facilitate cell type specific investigation. So switching gears here, we trained mice on an auditory discrimination task with delayed licking responses. So on each trial, mice were presented with auditory click trains of different rates. Mice need to discriminate the click rates, form and maintain a motor plan during variable delay period, and respond by licking one of the two lick ports after an auditory go cue. So low click rates means go left, high click rates means go right, and we counterbalance that across animals. So here we actually varied both perceptual difficulty and delay duration on a trial by trial basis, which allows us to examine the mechanism for motor planning with varying degree of memory demand. And you can see here that as delay duration increases, animals' response decrease, consistent with a motor preparation process. And in the past 8 years or so, converging evidence in HEFIX mice have indicated the secondary motor cortex, or M2, or ALM, as a cortical substrate for planning directional licking responses. So SC has not been studied much in the context of licking. So early on during our project, we conducted anterograde tracing from M2 projection neurons and found dense M2 terminals in this lateral part of the superior caliculus, suggesting its potential involvement. And we then conducted functional studies to test lateral SC's role in memory-guided directional licking. And to continue the research questions I presented earlier, we aim to characterize task-relevant signals in the M2 SC pathway and the causal requirement of this pathway for behavior. First, I optogenetically inactivated unilateral SC during the intertrial interval stimulus delay and response epoch. Shown here are the stimulus, delay, and response period inactivation. The red are the opto data, black show control performance. So we found that silencing SC activity resulted in a significant shift of the psychometric curve, which translates to a contralateral impairment and ipsilateral bias. In addition, the greatest impairment occurred when SC was inactivated during the delay period. So these inactivation data show that SC is important for memory-guided directional licking, suggesting a more general role of SC in spatial decisions beyond saccades and orienting, which are most of the studies that we look at SC. But if you're a grumpy reviewer, you might say that, well, SC is on the way from M2 to brainstem. So even if it just plays a very passive role, relaying the signal from M2 to brainstem, because it's on the way, if you inactivate it, you might still see an effect. If that's the case, then inactivating M2 and SC together should have a similar effect as M2 inactivation alone. In contrast, if SC plays a more active role that facilitate motor planning, simultaneous inactivation of M2 and SC should lead to additional behavior deficits. And that's what we tested here. And we found that inactivating M2 and SC together, not only did we find a larger behavior deficit overall, we also found a difficulty-dependent behavior impairment, shown here, after delay period inactivation, suggesting that M2 and SC make concurrent contribution, and maybe specifically to decision maintenance. So these inactivation data inspires us to ask, does M2 send choice-related signals to SC during different task epoch? To answer that, I did projection-based 2-photon Kells imaging from a subset of M2 neurons that project to the SC with retrograde expression of GCAM6. So SC projecting M2 somas are super deep. They reside in the lower level of layer 5B, which is, like you can see, quite deep. So the data I'll show you today include trunk as well as somatic data, which are usually highly correlated. Okay, so let's first look at a representative neuron. Here, each line is a trial. The color represents amplitude of Lorentz change, delta F over F. Activity is aligned to the goal cue and sorted by animals' response times, which are the red dots. And this neuron has higher activity when animals look contralaterally to the image side, and choice activity emerged before the goal cue and peaked around answer time. But of course, we saw other kinds of neurons, purely motor neurons or multiplexing neurons that had different information during sensation versus action. So how do we quantify the contributions of activity across neurons and compare different task epochs fairly? For each neuron, we extracted the appropriate frames per epoch per trial. Then we calculate the mean activity in that epoch for that trial and quantify choice activity across trials. So this way, we don't have to rely on any sort of alignment of activity across trials, which is bound to be unfair for one of the epochs because we have these variable delay and variable response time epochs. Okay, so this is the resulting simplified version of the fluorescence trace and PACH for the neuron that I showed you in the previous slide. Then I can conduct ROC analysis, for example, to discriminate left versus right choices for all 5 epochs in a fair way. For each epoch, I can look at the distribution of AUC values for the entire population. For example, in the sound period, there are only a few significantly modulated neurons in this example animal, and the distribution does not seem to show an overall control preference. In the delay period, however, more neurons start to show high contrast selectivity, and this trend continues during the response and the late epoch. You can see that the M2-SC pathway is progressively more contrast selective as the trial unfolds. In contrast, I should mention that when I image all layer 5 neurons with no projection specificity, I did not observe such a trend. So this is really seemed to be special for the M2-SC pathway. So what about on the real population level? Again, we used cross-validated linear classifiers to decode the amount of choice or sensory information during different epochs. Similar to single neuron analysis, population analysis showed that M2 sends progressively stronger choice-related signal to the SC. We then examined the relationship between the amount of choice information in this pathway and animals' response time on a trial-by-trial basis, using leave one out cross-validation. As I showed before, shorter response time tend to happen on trials with longer delay durations, consistent with the motor preparation process where movements are faster when more time is given to prepare for them. So if the delay period activity in the M2-SC pathway are important for motor preparation, which I've been talking, I think it is, the strength of the choice information should also be negatively correlated with RT. And that is exactly what we found. And we simultaneously quantify the effects of delay duration and delay neural activity in predicting RT and found significant effects of both. So these delay signals are important for animals' subsequent behavior, both choice and response time. But are these signals required for the behavior? Like I mentioned before, due to the interconnected nature of the network, a direct test of this hypothesis turns out to be quite tricky, right? For example, inactivating one central node could actually be equivalent to perturbing the majority of the circuit. So instead, what we want to do is to selectively inactivate one pathway while sparing the other projections. So we express HM4D in M2 neurons and locally infuse CNO in bilateral SC via implanted cannula, therefore only silencing M2 terminals in SC chemogenetically without affecting M2 activity in other projection pathways. And remember, we can assess the effects in trials with different delay durations and different perceptual difficulties. So what we found is that blocking the information flow from M2 to SC selectively impaired performance on heart trials with long delay durations. This difficulty and delay dependent effect suggests a critical role for the M2 SC pathway for decision maintenance, consistent with the multi-site opto experiments that I showed earlier. This is also important. I should mention that when I inactivated M2 somas with no projection specificity, I did not observe such a difference in effects. So in that experiment, the easy and hard trials were equally impaired. Okay, so for this part of the story, we found that SC is important for planning directional linking responses, extending its function beyond saccadin orienting. We found that M2 sends choice signal to the SC, and this signal during the delay predicts animals' choice and RT. The projection-specific perturbation results suggest that first, the role of M2 SC pathway cannot be compensated by direct M2 brainstem projections, and more cognitively demanding processes may require M2 SC cooperation. Finally, we ask, how are decision signals from M2 further processed within SC local circuits? And to address that, we first ask, what kind of SC neurons receive M2 input? We combined anterograde transsynaptic viral tracing with immunohistochemistry and found that M2 terminals target both excitatory and inhibitory neurons in the SC almost half-half. To conduct functional characterization of M2 SC synapses, we measure synaptic transmission in genetically labeled excitatory or inhibitory SC neurons using patch clamp recording, while optogenetically stimulating M2 terminals in these brain slices. So here are two example traces showing the synaptic potentials in example excitatory and inhibitory SC neuron in response to activation of M2 terminals. Again, confirming the existence of these different types of synapses. But of course, we want to know what these neurons are doing during behavior. We recorded population averaged calcium signals from either the excitatory neurons or the inhibitory neurons using fiber photometry. So this is without cellular resolution, right? Unlike the green blends that Ian has mentioned before in his talk, this is population average signals. We used different transgenic mouse lines, and we found that both populations show choice-related responses during the stimulus and the delay epochs, as shown here as in the example sessions. So for comparison, we see that in the excitatory example session, contra-selectivity is maintained over time. But in the inhibitory session, we see this switch in choice preference from early to late period. So to quantify that trend, here we plot on the x-axis choice preference during the early period, the stimulus period, and on the y-axis choice preference in the late delay period. So here, if a session go into the gray quadrants, that means they have switched choice preference from early to late period. And they're in the white quadrants, that means they maintain the same choice preference. So orange will be the excitatory session and gray will be the inhibitory session. And you can see that there are more gray/inhibitory session residing in the gray quadrants. So these suggest that the excitatory population tend to maintain the same choice information over time, whereas the inhibitory population display more dynamic and heterogeneous choice coding. What's really interesting is that these functional differences between the two cell types actually parallel the differential connectivity patterns of excitatory and inhibitory population. So the excitatory population, they project to the contralateral SC, but they also send ascending projection to the thalamus and descending projection to the brainstem nuclei. Whereas the inhibitory population, they only project to the contralateral SC. Of course, there are also local projections, but they avoid the brainstem and thalamus. So together, these cell type specific analyses in SC suggest that the contraselective motor planning signals from M2 are more faithfully maintained and transmitted to downstream regions via SC excitatory neurons, while the inhibitory neurons probably play more heterogeneous and modulatory functions within the SC. So putting it all together, our rat and mouse study suggests that the midbrain SC and evolutionarily conserved sensory-motor structure is also important for representing internal states, information that bridge sensation and action and are used to guide flexible behavior. And it plays an active role in cognitive processes such as inhibitory control, context-based vector inversion, and choice computation. One of the major inputs to the SC, the frontal cortex, sends task-related information to SC. And this information is modulated according to task demand, either pro-anti or more difficult versus easier choice during the delay period. And it predicts animal subsequent behavior. And this cortical calicular pathway may be specifically recruited for more cognitive demanding processes in a cell type specific manner that we're now starting to unveil. So with that, I'd like to thank my past funding support and my collaborators on both projects, and also like to thank my current funding support and a plug, advertise that the Duan Lab at SWC is hiring. We use rodent models of economic decision-making under risk, uncertainty, and social influence to probe individual decision preference and the underlying neuromechanism. So if you're interested in these, you should get in touch. And finally, thanks to all of you for your attention.
Thank you very much. Really impressive combination of techniques and modeling there. So do we have any questions in the room? Ian?
When you say the M2 to SC is recruited when it becomes sort of more cognitively planning, do you have an idea of what occurs during that recruitment process? Because obviously preparation to movement can occur just through the ALM to brainstem itself. Do you have an idea of what actually recruits that pathway and how it understands that it's more cognitively challenging, I guess?
Did you hear that, Anne?
Kind of. So the general framework, I feel like I missed the key part of the question. Okay.
So that overall it was do you have an idea about what recruits the M2 to SC pathway? Is that
it understands it's more cognitively challenging.
How does it get the information that things are more cognitively challenging?
Right. So first, like what I mean by cognitively more involved processes, I meant, for example, it seems like when we specifically perturb this pathway, the trials with long delay durations and the trials with more perceptual difficulty are more impaired. So from a behavior perspective, that's what I meant. Maybe that's what Ian's asking. There's a more interesting question of like how to implement that by the brain, right? What are some of the circuit mechanisms that actually recruits this pathway specifically for these processes? So one of the idea is maybe is related to how redundant the pathway is, the whole network is for different processes. Maybe for the easier, very short delay duration and very easily discriminated perceptual decisions, the M2 brainstem pathways are totally sufficient. However, while the difficulty increases, that's when the brain recruits more pathways. So you need more, and that's when if you specifically perturb here, then behavior is impaired. So I don't know if that answers. And also like SC obviously receive other input from for example, auditory cortex. So SC can also integrate different inputs, right? M2 is only one of the inputs. So maybe for these more, either for the anti versus the pro trial or for a large memory demand, what we have is a more kind of active participation of the SC itself.
Thanks. Any more questions? So I found your approach very interesting in the first study, that you really tried to use the modeling to establish the connectivity patterns. Have you or anyone else looked for whether there was an anatomical substrate for that?
Yeah, so that's the point of modeling, right? You want to generate some hypothesis to test. We haven't tested that ourselves. That's, I think, the obvious question. So thank you for asking that. So it's a very hard thing to do, right? Because I think now it's feasible. So what you want to do is to pick these neurons based on their functional tuning, and then do maybe either perturb these neurons specifically, and then look at activity patterns, or you have to do some crazy sort of transgenics to look at whether the inhibitory versus excitatory connections exist based on these functionally defined neuropopulations. So the short answer is that we haven't done that, but I think that was kind of the point that Carlos wanted to really pursue this modeling approach, because we want to generate some testable hypothesis, but it's a very hard hypothesis to test.
Yeah, I mean, it's a very interesting approach to produce the hypothesis and then look for the anatomy.
Yeah.
Anybody else? Okay, well, we're bang on time then, so I think we'll say thanks again to Anne. So welcome back, everybody. So I'm going to give the next talk, and I'm going to change tack slightly. So you've heard two brilliant talks about how specific networks of neurons encode actions through their changes in rate. I'm going to tell you a bit about how oscillations might be involved in movement, and in particular in disorders of movement. So this rhythmic activity is a core property of neuronal circuits. And so, we, of course, there are these kind of central pattern generators as well in mammalian brains to control, again, locomotion and breathing. And then you can also see in the human brain a really extreme example of rhythmic activity in people with tremor, either essential tremor or PD, when you have these very rhythmic bursts of activity in muscles, which make the limbs tremor. And you can see corresponding activity that's driving this activity in the muscle in the motor thalamus. So, of course, in the cortex particularly, but all over the brain, there are slightly more subtle oscillatory activities, and these can be recorded with various methods such as EEG, ECOG or electrocorticogram, and LFP local field potentials. So here's just a summary figure from this review by Bijaki, where they're showing that you can pick up these rhythmic oscillations in these different cortical signals, but essentially you're always picking up the same thing, and that is underlain by synchronized synaptic activity, which summates particularly well in cortical columns. And so when we're looking at these oscillations in EEG and MEG, what we're really looking at is summated membrane potentials across neurons, which build up to something you can see in a local field potential or an EEG. So in the motor cortex, a particularly prominent oscillation is in the beta range. And this has been described many, many times in many different experiments. One of the classic ones was this study by Murphy and Fett in '96. So they recorded with depth electrodes and primary motor cortex of animals performing simple motor tasks. And what you can see is these great big ripples of beta activity. So somewhere between around 12, 15 and 35 hertz. And you can see these particularly clustered in the motor cortex. And we often measure the strength of oscillation with a power spectra, whereby we have frequency here on the X, and then the strength of the oscillation on the Y. So here you can see this big peak around 30 hertz. And then they also show, when they recorded single units, that these big ripples of beta oscillations are able to time action potentials in individual cortical neurons. So you can also see prominent beta oscillations in EEG and MEG signals in humans, and these experiments, I could have picked any one, there's hundreds of them, showing these very robust changes in beta oscillations in response to movement. So here we have a simple finger movement. The beta is now shown by this color scale frequency on the y-axis, and you can see as you approach the movement offset, you get a beta desynchronization and then a beta rebound. And these authors here were able to beamform and show that the source of this was in primary motor cortex. And so this experiment has been done over and over again, and you always seem to get this beta desynchronization around the time of dynamic movement. So why might these oscillations be useful for motor output? Well, again, what they seem to correspond to, so here now we're looking at EEG and muscle in human participants. You get this big increase in beta activity at the point where there's a kind of holding of movement. So here we've got a precision grip task where you have an apparatus that the subjects just have to grip and hold and then move to another hold period. And what you see is this big increase in beta around the time of the holding. And you can also see this in the EMG, so or the muscle activity. So you seem to have this, maybe that this transmission at beta frequency helps muscles hold a particular position. This is another example where this was Pascal Friese's group, and what they did was they looked, they stimulated using the motor cortex using TMS, and then they did a post hoc processing of where this TMS pulse was given on the phase of the EMG oscillation. And what they saw was that the different phases predicted how big the motor evoked potential was in the thumb muscle. So it seems like where a given input it lands on the phase of a given beta oscillation will change how much power is given to the muscle. So another big source of work on beta oscillations is patients with Parkinson's disease. So I'm sure many of you know that there are the cardinal symptoms of Parkinson's disease are an absence or slowness of movement, akinesia and bradykinesia, stiffness and rigidity, and then a subsection of patients also have tremor. So many things happen to the brain in Parkinson's disease, but the kind of defining feature is a loss of dopamine in the striatum due to a death of neurons in the substantia nigra pars reticulata. And this affects a network of areas called the basal ganglia, as you've already heard mentioned today, which form loops with virtually all areas of the cortex. But in Parkinson's, it's the motor cortex, which is likely to be particularly involved. So Peter Brown, who until he recently retired, worked here in Oxford. Over a period of about 20 years, he's described beta oscillations in Parkinson's disease. And his original insight was that in patients who receive deep brain stimulation electrodes to treat Parkinson's disease, where you put in a constant current at 130 hertz, there's a window between the stimulator being implanted and the electrodes, and you can plug these electrodes into an electrophysiological acquisition system and record the local field potentials from the subthalamic nucleus. And so what Peter saw was these florid beta oscillations in the local field potential. And of course, when he first did this, he didn't know whether this was just a healthy activity or something to do with the disease. So he compared the power of these oscillations in patients on and off medication. And what he saw was only when they were fully Parkinsonian, off their dopaminergic therapy, do you have this big peak in the beta range, which is completely lost when dopamine is restored. And also that the strength of the oscillation correlates with the severity of akinesia and rigidity. So another important feature then, going back to cortical beta, is that when you record EEG in these patients together with their local field potentials, you see that beta oscillations in the cortex are highly synchronized with those in the subthalamic nucleus. So we have this specific synchronization at beta frequency between cortex and basal ganglia. So but just to reiterate then about the basal ganglia, so this network of subcortical structures. And in Parkinson's disease, we lose this dopaminergic regulation of the striatum. So obviously this ability to record in humans is amazing, but is limited to wherever the electrode is going for therapeutic purposes. So what a lot of people have wanted to know then is whether these oscillations encompass the entire network. And so to do this, ideally you need an animal model. And during my PhD with Peter Brown, we thought that rats, the 6-hydroxydopamine lesioned rat, where you kill the neurons in the substantia nigra, resulting in this loss of dopaminergic signaling to the striatum, might be a good model for the disease. And what we found was that in these animals, both in the subthalamic nucleus local field potential and in the ECOG, we could see these big beta oscillations, which really resemble quite closely those that you see in patients. And when we looked at the coherence, so the coherence is how much does the phase of the oscillation in one structure predict another, we see this massive increase in coherence between EEG and SDN only in the lesioned animals in the grey. So Pete McGill, who's also here in Oxford, he really used this over a number of studies, including some of my own, to look then at different populations of neurons in the basal ganglia and how those individual populations of neurons lock to cortical oscillations. So a lot of this work was using silicon probes placed into the globus pallidus, subthalamic nucleus, striatum and thalamus, and then looking at how these spikes related to the beta oscillations. So to summarize really an awful lot of work in one slide, what you can see then is if you look at, if this represents the idealized beta oscillation that you record in the cortex, you see that different populations of neurons, in this case, the indirect pathway neurons in the striatum, which become hyperactive in the disease, start to lock to a specific phase of the ongoing oscillation. And when you look through all the different structures in the basal ganglia, you see the same in every single structure. So each population has a preferred phase that the spikes in that structure seem to lock to. So in some ways, and although I haven't shown it, you see very little locking in healthy animals. So what Parkinson's disease seems to do almost is turn the whole basal ganglia network into a sort of pattern generator, where everything becomes locked to those beta oscillations. And then in turn, because the basal ganglia project through their output nuclei to the thalamus and back to the cortex, this rhythmic activity is going to reinforce the cortical oscillation. So I've given you a bit of an outline of beta oscillations in healthy and disease contexts. And so Peter Brown and Simon Little kind of put this theory across that there's a sweet spot of beta synchronization which helps to maintain certain motor behaviors, perhaps like holding, perhaps to increase transmission through the corticospinal pathway. If this becomes too high, you have a very high signal to noise ratio, but basically you lose all your information in the signal. So you kind of ideally want this trade-off where the oscillations are useful, but don't reduce all the information by synchronizing everything. So just to summarize then, cortical beta oscillations are reliably modulated by specific aspects of movement, but become highly synchronized in Parkinson's disease. So what would be really useful would be, in order to really probe the role of these oscillations, would be to be able to control them bidirectionally, to either suppress or amplify them in the same animal. And so for the rest of the talk, I'm going to talk about whether it's possible to harness temporal dynamics of the oscillation to suppress and amplify. So what do I mean by temporal dynamics? Well, if we think of the oscillations in terms of a pendulum, so it's a very simple analogy, and like all simple analogies, doesn't work perfectly, but I think it's quite illustrative. So I stole this figure from a paper that's actually about oscillations in speech. So you could think of the pendulum only being moved by an external input, in this case a motor. So this might be like if an oscillation in the brain was driven only by electrical stimulation or by rhythmic sound or something, but had no capacity to carry it on itself. Then you can have a passive oscillation whereby if you push the pendulum a few times, it will build up in power, and then as the power dissipates, it will naturally decrease again. And this is a bit like what we see with beta oscillations in the brain. So they occur in these bursts where they seem to build up in power and then naturally go again, then the whole process starts again. So then if you think of an active oscillation like a child on a swing, if you apply energy at the right time, you can propagate the oscillation and make it stronger and more stable. And so our kind of core hypothesis was that maybe if we could deliver a perturbation, in this case electrical stimulation, on specific phases of the network oscillation, we might either be able to speed up the dampening and reduce the power of the oscillation, or to provide this input like a child on a swing and increase the stability of the oscillation and make it propagate. So in order to do this, a postdoc in my lab, Colin McNamara, designed a digital circuit which was capable of tracking the phase of an ongoing oscillation. And in this case, I'm always talking about the ECOG recorded in 6-hydroxydopamine rats, which is a particularly strong beta signal. So I could probably give a whole talk about the problems with designing this circuit and making sure it worked. So for the purpose of this, you'll just have to take my word for it that his circuit is really good at tracking phase. And so what it can do is instantaneously track the phase of the ongoing cortical oscillation and use that phase then to turn it into a stimulation train, which will deliver stimulation back at a specific phase of that oscillation. So in this case, it would be the peak. And then what we're going to do is measure how that perturbation affects the power of the ongoing cortical signal. So what's particularly special about the way that Colin did this is that he can embed it on a programmable circuit within our recording system. So the cortical signals come in, the processing is done instantaneously in order to deliver the stimulation output. And when you're working on timescales of around each cycle being 30 milliseconds, this latency really matters. If you were to go in and out of a PC, you'd have too much delay. So we chose then to record the cortical signal, track its phase and deliver stimulation into the GP at specific phases of that cortical signal. And we did that because the GP seems to be a really key node in driving beta oscillations across the entire network. So our hypothesis then is that if we deliver stimulation on specific phases to the GP, we'll make those spikes even more tightly bind to their specific phase on the cortical oscillation, and that will in turn drive higher synchrony across the network, eventually ending up in a bigger, stronger signal in the cortex, whereas other phases will do the opposite, break the spikes away from their preferred phase, and eventually reduce the oscillation in the cortex through the circuit. So a lot of the experiments here were done by a student in my lab, Max Rothwell, working with Colin. So a lot of the recordings I'm about to present were all done in an open field. So there are awake rats with chronic implants where we're recording the ECOG from a screw over the motor cortex and delivering stimulation with another electrode into the GP. So what we did was we would record the beta oscillations for a while, and then we would give epochs of stimulation at a specific phase of the cortical oscillation, and we'd basically cycle through 8 different phases. And so this is just to show the beta oscillation in the baseline periods in brown, and that it's pretty much the same in these intermittent periods. So when we turn the stimulation off, it's just like it would be if we recorded for a long time before the experiment. So here you can see some raw traces from one of these experiments. So in the black, we have an animal with no stimulation. You can see these ripples of beta oscillations. Below is just the filtered signal between 30 and 40 hertz. You can see them waxing and waning. So in this orange trace now, we're stimulating on the GP on the descending phase of the cortical oscillation. And hopefully you can see very clearly that the oscillations are now being propagated and that the whole amplitude of the oscillation has gone up. So it looks like we're managing to amplify. If we flip to the other side of the cycle, we have completely the opposite. So now we've got lower amplitude oscillations and they're less stable over time. So if you take the most amplifying and suppressing phases and plot the power spectra, in this quite large sample of rats, you can see that we can really reliably amplify the beta oscillation and suppress it as compared to the control oscillation. And just remember, this is in the same animal. So we're able to flip between amplifying and suppressing in the same animal. And what was quite nice was that there was a real consistency in the phases that were amplifying and suppressing. So this suggests that, so here we have the change in beta power on the y-axis and the suppressing amplifying phase on the x, and you can see that the amplifying and suppressing really cluster to this first part of the descending phase and first part of the ascending. So it looks like we're really doing something consistent to the circuit every time in order to amplify and suppress the oscillation. One of the things that we found really interesting is that, so the way that Colin programs his circuit, if the phase estimate becomes unreliable, so if the oscillations become really low in power, his system will detect it and not stimulate. And this guy gives a kind of second closed loop element whereby if you amplify the oscillation, you're more likely to stimulate because the phase estimate becomes more reliable and vice versa. So this is 5 different periods where we start to give either the amplifying or suppressing. So you can see in the amplifying that we almost immediately start to boost the beta oscillations. And also the stimulation train, which is these black marks, is really quite relatively regular and goes on these long trains of regular stimulation. Whereas the suppressing is much more irregular and we have big gaps because we're reducing the phase consistency. And so if you plot the autocorrelogram of the stimulation train, so this is how much can you predict the following stims from any given stim at time 0, what you see is that the amplifying is highly regular, whereas the suppressing is not, gets dampened very quickly. So I think we've got reasonable evidence that we are able to kind of dampen down like these passive oscillations and suppress an oscillation, or when we apply energy at the correct time, we can increase its stability and propagate it. So this is a symposium about motor control and action then. So what happens to the animal's behavior? So we decided to test this on this commercially available catwalk system, which is basically an infrared camera, which is placed under a platform, and the rat runs back and forth across this platform to get a small reward at each end so that we can get lots and lots of trials. And then we can measure the gait of the animal and its speed as it keeps walking along. So Max and Colin did a heroic job of modifying this piece of equipment so that you can actually perform closed loop stimulation on a behaving animal. And this is just to prove that we could have the same kind of effect. So here, this is when the animals are behaving, we can get this great big amplification and suppression in the yellow. Here you can see the mains noise, and you can see that all our beta oscillation modulation is very separate to that artifact. So what we did then was to compare various motor parameters between amplifying and suppressing and also in baseline. And what we saw was that animals would run across the platform at very different baseline speeds. So some of them will kind of amble along getting their rewards, and others, showing that perhaps our lesion wasn't particularly severe, could really go quite quickly across the platform. But what we found was whatever the baseline speed, during amplifying stimulation, the speed was lower, and during suppressing stimulation, the speed was higher. So this is broadly consistent then with beta being kind of anti-kinetic and slowing movement as it does in Parkinson's disease. So we kind of wanted to know then, so this is just, you can see 2 animals walking across the catwalk system. We wanted to know what exactly happens. Why do they get slower? Do they run in exactly the same way, in a slower way, or are they actually changing the way that they move across the platform? And so we hadn't really realized that rats have 2 very different styles of moving that they kind of alternate between. So they alternate their limbs in a very classical walk, but they can also bound a bit like a cheetah, where they'll put both paws forward and then bring the back paws up. And so what we found is that our changes in speed were almost completely predicted by how much the animal was bounding and how much it was walking. So walking is much slower than bounding, as you can imagine. And basically what we found was that as the speed reduces, so the blue dots here are the suppressing and the orange dots are the amplifying, that you get a basically linear reduction in bounding to walking. And so this could really, this change in the bounding could really explain really most of the variance in the actual speed. So we found this really interesting. So the beta wasn't just slowing ongoing movement, it actually seemed to, by amplifying it, it was changing the bias in the entire way that the animal was moving. So what you might say then is, well, maybe the closed loop isn't necessary, both for the physiological and behavioral effects. Maybe it's just that you're producing a pattern of stimulation that's different between suppressing and amplifying. Because of the way the closed loop works, as I said, amplifying is much more regular than suppressing. So what we did to kind of counteract this confound was to perform a set of experiments where we would record in a closed loop, and then we would take the stim train from that closed loop experiments and directly afterwards play it back into the GP open loop. So the pattern is maintained, but there's now no relationship to the cortical phase. So what we found was that we still got, so in the closed loop, we reproduced our results from our core experiments. And we found that in amplifying, we still got a little bit of amplification. And this is perhaps not surprising because you're putting in quite an oscillatory stimulus, but nevertheless, it was significantly less than the closed loop amplification. What was really interesting was that when we played back the suppressing stimulation train, not only do we not get suppression, we get amplification. So this shows, and you can see this summarized here, each dot being one recording. This shows that the closed loop was absolutely crucial for suppressing the oscillation. And in fact, without it, the same input would amplify it. So just to summarize these rodent experiments then, so cortical beta oscillations synchronize activity across the basal ganglia, but it is possible by adding energy in the right place to suppress or amplify these ongoing oscillations, and this seems to bias the animal's locomotor pattern. And in the case of physiology, we're able to show that this is really dependent on forming a closed loop. So I just wanted to very quickly show you a different study, which actually we managed to complete before the closed loop one, which was trying to establish whether these principles would actually hold in the human brain and specifically in Parkinsonian patients. So this was done in collaboration with a group in Hamburg, headed by Christian Moll, where I did my postdoc, and two former members of the lab, Abby Holt and Esther Corman. So what we wanted to do was, we knew we weren't in a position to try and deliver closed loop in human Parkinsonian patients. But what you do have is the opportunity to deliver stimulation in the operation to implant DBS electrodes. So here you can see a picture of one of these operations, and in the back you can see our acquisition computers. So what they do in Hamburg and in many surgical centers is use microelectrodes to map the SDN area to make sure they're going to put the therapeutic electrode in the right place. So you map with these 3 electrodes, which have a microelectrode, and then 3 millimeters above, they have a macroelectrode where you can deliver stimulation. So it's possible to record a local field potential from the subthalamic nucleus while delivering stimulation above it. And this is a slightly dirty experiment because you're stimulating the fibers from cortex and GP that go into the SDN. You're probably stimulating SDN a bit. So it's a kind of slightly non-specific perturbation. So what we tried is that, what we kind of hypothesized is if we could deliver stimulation in at these macrotips at the frequency of the patient's beta oscillations, we'd have periods where the stimulation would hit the same phase over and over again because the 2 frequencies would be matched. And just to say, so basically through a specific stimulation configuration and some clever artifact removal by Abby, we were able to rescue the signal then and see where the stimulation landed on the ongoing beta oscillation. So here you can see an example then. So we've split the phase cycle into 4, and we get these runs of stims that will land when the stim cycle is cut into 4 on 7, 8, 9 of this, basically the same phase in a row. And then there'll be a slight change in frequency or power, and that will basically flip the stim onto a different phase. So then what we could do is afterwards, we could look at where the stimulation landed on the oscillatory cycle, and we could say, is that power of that signal increased or decreased as you get consecutive stimuli on a given phase? So here in this example, you can see on the blue phase here, once we hit 3 in a row of this descending phase, we start to lower the beta amplitude. And then if we flip to the other side of the cycle, we start to amplify it. So it's exactly the same principle as in the closed loop experiments in the rats, and shows that it may be a feasible way of modulating oscillations in the human brain. So just very quickly then, I wanted to highlight that this approach goes a long way beyond Parkinson's, because of course there are oscillations associated with many different behaviors and many different diseases. And one of the projects that we've got is in collaboration with Charlotte Stagg, where she has this very nice paper on bioRxiv, where she demonstrates that different configurations of alternating current stimulation over the motor cortex, where she delivers gamma oscillations at different phases of an underlying theta oscillation, can modulate a simple motor behavior where people are trained to go faster and faster acceleration between their thumb and index finger. So what she sees is that this specific configuration increases the learning rate over this task, and that actually goes beyond the end of the stimulation period. So she's able to kind of really produce a quite striking increase in motor learning speed. And so what we're doing is saying, well, maybe we can do even better if we deliver the gamma on the intrinsic theta oscillation that you see in the motor cortex. So this has been implemented by Jess Meyer and Robert Toth in my lab. And so what they've managed to get working is delivering pulses of gamma oscillations on specific phases of this ongoing theta oscillation. And they're delivering optogenetic stimulation to parvalbumin neurons in the motor cortex. And Charlie's hypothesis is that the PV neurons are particularly important for plasticity. So this project's in its early stage, but what we've been able to do is already show that using this setup, we can either suppress, just like in the Parkinsonian animals, the theta oscillation or enhance it. And so because of the way that this has been implemented by Colin, there's really a lot of translational scope for getting these techniques into humans. Because this is a digital circuit, it can be implemented fairly directly into the new generation of closed loop devices, which can handle these kind of circuits and sense as well as stimulate. And also what we're very interested in is doing feedback experiments for non-invasive stimulation, where we might record an oscillation in the EEG and stimulate on a specific phase of that using something like TMS. And this is all in collaboration with Tim Dennison in the unit. So I'd just like to particularly acknowledge Colin McManara and Max Rothwell, who did all the rodent experiments, which have really taken quite a long time, and to all the other current and past members of my lab who also contributed to these projects, and to my funders. Thank you. Any questions? Ian?
What are the main challenges you face with switching to this from sort of standard frequency stimulation? This sort of phase oscillation?
In the clinical sense?
Yes.
I mean, I think one of the biggest challenges is dealing with the stimulation artifact. So, you know, we got round this partly by using 2 different parts of the network, but if in DBS, you'd have to record on one contact and stimulate on a neighboring contact. So you've got to make sure that you're not just in training to your own stimulation artifacts. That's definitely one of the biggest challenges. I mean, certainly though, you know, the actual tracking can be handled by these new devices. And Tim's implant that he's working on is particularly interesting because it's mounted on the skull. So you get rid of a lot of the artifacts which might cause problems with your sensing, for example, ECG artifact.
And you think the technology will be dynamic enough to, obviously the symptoms of Parkinson's are quite different times in different patients. Do you think it'll be dynamic enough to be able to train to what that particular patient's?
Well, basically, ideally, you would have another loop which dealt with things like the sleep-wake cycle, whether someone was moving or not. So you kind of have loops on loops,
yeah, because, you know,
and activate different protocols at different times.
Sitting tremor versus
Exactly. Yeah. Masood?
Very interesting. So is the oscillatory behavior an emergent property of the entire network, or are there critical nodes in this network? Because you mentioned pattern generators.
Yes, so I think it's a difficult question to answer. So in general, the clinical and experimental evidence is that if you lesioned any node, you could probably get rid of the oscillation. But then there are parts of the network that seem to be specifically important. So GP, for example, projects to so many other structures that it's probably particularly important for really the timing becoming very synchronized across the whole network. We just published a paper, Koichi Nakamura in Pete's lab just published a paper showing that if you inhibit thalamus, the cortical oscillation stops almost immediately. So you do need this feedback loop round back to cortex. So the short answer is everything is important, but probably for different reasons. Yep.
So I just have a short question following from that. I just want to make sure I got it right. I remember food flies are actually like pattern generator cells that generate these patterns. This is quite different.
Yeah.
The pattern is not being generated, but it's in the circuit itself. So
yeah, I mean, apologies. So really all I was trying to illustrate with the pattern generator is an extreme example of rhythmic activity being generated by a circuit. I mean, oscillations in the forebrain are much more subtle in the way they're produced, and you know, kind of there are both local and circuit mechanisms to generating them. So yeah, they're a bit more complex than the kind of true CPG that would control locomotion. Okay, well, in that case, I think we'll move on. Thanks everyone for your questions. So our next speaker is Claire Press. So she works both in Birkbeck University in London and for the Phil at UCL. And she's going to change our trajectory again a bit and tell us about how action and perception interact to affect movement action. Thank you, Claire.
Thanks very much. Yeah, so just to introduce myself, so I'm head of the Action Lab, Birkbeck and UCL. Lots of our questions, we're called the Action Lab, but we're very focused on how action interacts with perception. Of course, it's crucial that we understand perception as action researchers, and it's all human work I'm going to be presenting to you today. So, of course, all action researchers would find it indisputable that perception is playing a very central role in our control of action. So that is the function of our actions is to interact with our environment. Of course, we must perceive that environment. So, for instance, when we're wishing to move towards the cup, It's essential that we can see where this cup is and to monitor our actions online as we move towards it. As we wish to move away from a hot stove, that has been triggered by a sensation of the pain from which we are moving away. As we converse with a friend, we are hearing their speech, we're seeing their gestures and performing appropriate actions in reaction to that stimulation. So it's essential that we understand then how the motor world interacts with the sensory world. You might then think that the, of course, perfect scenario for effective action control is that we perceive the entirety of our dynamic, ever-changing environment, and we perceive it perfectly and then act upon that information. But of course, what we all know is that this is an impossibility. So we have a multitude of sensory information bombarding all of our sensory receptors, sights, sounds, smells, changes across time, changes across space, basically repeatedly. There's no way that we can possibly be processing all of that. So we have limited capacity systems. We can only process some of these inputs. It's also the case that even for those inputs we process, there's a lot of noise that means we cannot process them perfectly. So there's external noise. So the cup we wish to grasp is partially occluded. The friend we're trying to listen to on the train is being masked by the sound of the train rattling around in the tunnel. And then the brain, of course, adds a whole host of extra noise. So we can't perceive our environment perfectly. What we need to understand is how we optimize perception within those constraints to serve action most effectively. And what many researchers working on this puzzle are fairly agreed upon is that in order to overcome these challenges, we need to use our knowledge about likely sensory events in order to shape what we perceive. So that is, we build up across our lifespan knowledge about statistical regularities, knowledge about what goes with what, and the types of sensory events that can follow from our action. So for instance, we can predict that when I go to grasp a cup, for instance, that I am making predictions about, okay, these motor commands are likely to lead to sight of the hand moving outwards. They're likely to lead to cutaneous stimulation on my fingertips at a certain time point in this motor command. And then it is agreed upon that we're using those predictions in some fashion to shape what we perceive. But this is then where the controversy starts and where lots of research is going into trying to figure out how exactly those predictions can be working to shape what we perceive. So what we are trying to optimize here, how this is feeding perception, and of course with the longer term view too to how this perceptual information is needed for shaping our subsequent actions. So theories that have been incredibly popular in motor domains for the last few decades are often called cancellation attenuation theories. But what they are proposing is that once you have predicted what is likely to result from sending certain motor commands, you are subtracting this prediction from what you ultimately perceive. So in some way attenuating that perceptual information or turning down the volume on those particular sensory channels. The reason that researchers have thought that such an adaptive function would be so useful is that if you turn down the volume on what you knew was going to be there on the basis of your motor commands, this is in turn allowing much processing of those unexpected events that we did not know were going to be there. So that is, we'll be more able to perceive them, we'll be perceiving them more intensely. And this is a very important thing for the organism to do because it's those unexpected events that are actually telling us something. So they're informative to the organism because by definition, we didn't know they were going to be there. So that means that actually our model of the world was incorrect. We need to use that sensory information to update our models. And if we're performing actions, very importantly, to be correcting our course of action. So the motor commands that we sent did not, in fact, achieve the desired outcome. A whole host of data that have been used to support these models over the years But to anyone working not directly in this field, the data that you're probably most aware of are the wide range of studies demonstrating across the last 3 decades that you cannot tickle yourself. Of course, everyone can appreciate this, as well as knowing that there are studies out there showing that. So what is assumed here is that when you send a motor command to move your fingers lightly across your skin surface, you anticipate what is likely to result from sending those motor commands, and you perceive those sensory consequences that are consistent with that expectation less intensely than if someone else was to unexpectedly tickle you. So these have been very popular ideas, as I say, across a few decades, partly due to the amount of data supporting them, but partly also because they do tell a very sensible story about how we can render the perceptual information within our environment useful to the organism. So that is, when we have the such limited capacity systems, it's ensuring that we have sufficient resources for processing those elements of our environment that in fact we need to do something with. Where it becomes problematic is when you consider what is being claimed outside of action communities, so from other people studying perception across the same time frame of a few decades. So if you look to what I'm going to call here the normative sensory cognition literature, on the whole here I mean people not studying action. So a large number of visual scientists studying how we process edges, objects, larger scenes, also tactile scientists, auditory scientists. The theories that have predominated here across the last couple of decades tend to be couched in Bayesian frameworks, and will be proposing something that is quite conflicting, in fact, with what's being proposed in the cancellation accounts. Because what's proposed here is that you predict what you're likely to perceive, and you combine that prediction with the incoming sensory evidence to determine what you perceive. So if you're familiar with these Bayesian frameworks, what this is saying is that you combine in a weighted fashion your prior with your likelihood to determine the posterior. And this type of process, in fact, leads to an upweighted rather than downweighted perception of what you expect. So that is, the expectation is directly feeding into what you perceive, which renders you more likely to perceive what you expect or perceiving it more intensely. So in fact, when you look at it like this is quite the opposite from with respect to the cancellation accounts. The adaptive functions focused on in these accounts for why the organism would need such mechanisms are quite different from those focused on in the motor community with these cancellation accounts. So they don't focus on the limited capacity systems and our inability to process everything, but rather on this sensory noise challenge facing our systems, which is that we're being bombarded with information that is incomplete, noisy, and we're trying to, on the basis of that noisy, incomplete information, generate experiences rapidly because our environment is forever changing. And so what is proposed here is that because this is so important to the organism, if we have this type of mechanism whereby what we expect is feeding into what we perceive, it will tend to make our experiences more accurate in the face of these challenges, because by definition, what we expect to be there is more likely to be there, assuming that your environment is fairly stable. So to illustrate the logic underlying these accounts, I'm going to show you these panels down here, which is a cartoonized description basically of the account. So if you consider that I'm just walking home one evening and I take a quick glance up the path in front of me, I might be presented with something that looks like this input panel here. So that is, it's dusk, there's not brilliant sensory information to be obtained, but also I've obtained it very quickly. So I haven't got a very good representation of really what is up in front of me. However, I do have an expectation that I should see my sons in front of me. And if I combine that noisy, quickly acquired information with that expectation, then this combination, in fact, provides a more accurate representation of what is out there than if I was to rely on these noisy inputs alone. So if you place these motor accounts next to these accounts popular outside of action, next to each other, they do seem to highlight a conundrum, which is these motor accounts are proposing these mechanisms that would do a very good job of optimizing informativeness of our experiences, which is very important for action control. But in doing so, they are making our experiences less veridical, and vice versa. So if you look at these the Bayesian accounts outside of action, these processes are doing a very good job in principle of making our experiences veridical, but they're directly acting against the informativeness of what we perceive. So this has been baffling us for the last few years concerning how we really can be using our predictions to shape what we perceive to serve our actions most effectively. As a starting point, we wanted to sit back and think whether action predictions really should be having a different influence on what we perceive relative to other types of prediction. So is there some different adaptive function in active settings relative to passive settings that means that we have different priorities, really, so that one type of challenge is more pressing relative to another type of challenge? And looking at those adaptive arguments, we, certainly couldn't see anything clearly that stood out there. So that is, of course, during action, we need our experiences to be informative. So that is, we need to be told about that stuff that means we should correct our actions or that we should update our models. But it is also absolutely crucial that we have a veridical representation of where we are in space, of where the objects are. These problems associated with sensory noise and incomplete information and rapidly changing environments are in fact more the case often in active settings than passive settings. There seems really no clear reason that this is less of a problem in active settings. So these adaptive arguments do not in fact appear to apply more to some contexts than others, or certainly along the dividing line where you find the different theories. It might then of course be the case that, so it doesn't immediately make sense, but we're scientists as well as philosophers, we have these decades of data that supports these mutually incompatible accounts, and therefore we do have different mechanisms. These accounts are different for a reason because they're based upon data. In fact, what we realized a few years ago when we started thinking about this was that the accounts are incompatible, but in fact the data are incomparable because very different types of data have been used to support these mutually incompatible accounts. So as a starting point to try to figure out how we're making our perceptual experiences useful during action, we wanted to ask whether there was any evidence that action predictions shape perception differently. So therefore, as this starting point, should we be trying to understand this as any problem specific to action, or should we be looking at models that actually apply in passive settings too? So for instance, trying to predict what will be in our environment on the basis of sensory context. So what we have been doing, and I'm going to present a few studies to you today, is to take the paradigms and analyses from outside of action where they have found support for these Bayesian accounts of perception and neural processing and take these to action. So do we see evidence for different types of process in action, or in fact, should we be looking at the same types of process in action and outside of action? And then actually we have some problems to solve concerning how we make our experiences veridical and informative regardless of setting. So I'm going to start by presenting to you some MRI studies that we've conducted, because in action, lots of the evidence used to support these cancellation accounts is coming from MRI findings of a lower signal in primary sensory processing regions when processing expected outcomes of action relative to unexpected. So it's been found across modalities really, but to give some examples, there's quite a few studies demonstrating a lower signal in primary somatosensory cortex when perceiving expected tactile action outcomes relative to unexpected. And it's been found that there's a lower signal in primary visual cortex when processing expected visual action outcomes. What we were especially intrigued about was that this is being used, this lower signal, to support the idea that we're just cancelling out these expected sensory consequences of action. We're basically turning down the volume on the sensory channels responsible for processing something that we expect to see or feel. But it was shown about a decade ago outside of the action literature that a lower sensory signal is not necessarily indicative of a cancellation type mechanism. Peter Koch in Floris de Longe's lab at the time conducted a study where they paired tones at different pitches with gratings at different orientations, so clockwise or counterclockwise relative to the vertical. And then they looked at the activity in primary visual cortex in response to those gratings as a function of whether the grating was expected or unexpected due to the preceding tone. And what they found was that regardless of the task that participants were performing on the gratings, judging their orientation or judging their contrast, there was a lower signal in primary visual cortex associated with the expected gratings relative to the unexpected gratings. So in fact, this finding is exactly the same as the finding that's been used in action to support these cancellation type accounts. The thing they found that is less consistent with a cancellation type mechanism is that if you train and test pattern classifiers on the basis of the signal in primary visual cortex, they in fact perform with better accuracy on the expected than the unexpected trials. So the signal is lower, but the pattern classifiers appear to have more information. They're representing this event with greater fidelity. So what might be going on? So if you consider that primary visual cortex has populations tuned towards edges of a wide variety of orientations, this blue line here is representing a hypothetical baseline of what you might expect these populations to be doing when presented with a vertical edge. So that is, the populations encoding the vertical should be more active than the populations coding the off vertical. What the cancellation accounts are proposing is that when you expect the vertical, that you're getting a signal reduction specifically in those populations encoding the thing that you're looking at. So you have subtracted what you expect from the sensory processing or turned down the volume on these expected channels. Consistent with the lower signal, but less consistent with this superior pattern classification of expected events. An alternative that is more consistent with the MVPA data is this sharpening account that Peter and Floris proposed, which is that when you expect a vertical edge, you pre-activate those units encoding the vertical, so the thing that you expect, and via a lateral inhibition process, you suppress processing in all other unit types. So this results in a sharper representation of that sensory event, which is consistent with the superior decoding, but also in terms of accounts of perception, more consistent with the idea that you're more likely to perceive what you expect or perceived in it more intensely relative to the cancellation accounts that are saying quite the opposite. So we wanted to know which of these was happening during action. And so Daniel Yon led these studies. So he was in my lab for quite a long time and has led a lot of the work that I'm presenting to you today. So what he did was to require participants to lie there performing finger abduction. So they would either abduct their index or their little finger. And as they did this, they would be observing an avatar hand on screen that was performing movements synchronized with what the participant themselves was doing. On congruent trials, they would observe the avatar moving the congruent finger. So that is, the participant has sent a motor command to move their index finger. And as they do this, they're seeing an avatar on screen moving its index finger. Or they can see incongruent movement so that they are moving their index finger and the avatar is moving its little finger. We are assuming here that congruent movements are more expected than incongruent movements on the basis of a lifetime's experience. So you have much experience of sending motor commands to move your index finger and seeing index fingers move. or certainly far more than doing that and seeing little fingers move. And then we performed multivariate and univariate analyses to try to understand the underlying mechanisms a bit better. The MVPA results basically are completely in line with these tones predicting gratings. So across a range of different visual ROIs, we found that the congruent expected visual action outcomes are represented with greater fidelity than the unexpected. So this is across bilateral occipital cortex, left occipital cortex, and right occipitotemporal cortex. We then looked at the univariate data, but not just doing this broad brushstroke approach that has been used in previous studies of looking for the global signal across the whole region. What we did instead was to separate the voxels according to whether they were what they were most responsive to. So then we could basically separate the analysis according to whether what's going on in the voxels that are especially responsive to the thing you're looking at, and those that are more responsive actually to other things. And what we found across the board was that in the units that are especially responsive to the thing you're currently looking at, there's no sign of a lower signal on the congruent expected trials relative to the incongruent unexpected trials. So that is, the saturated bars show no sign of being lower than the unsaturated bars. All of the univariate suppression is seen in the voxels that are preferentially tuned to other things. So this is much more in line with this sharpening graphic that I've shown you, whereby the signal reduction is coming from the off-center rather than the center. And perhaps most importantly for the state of play currently concerning our understanding of these mechanisms, more consistent with the findings outside of action, that, well, basically it's demonstrating that there does not seem to be something very different going on in action, as the models might have you believe. We've replicated this. I'm not going to tell you the twist that we performed because of time, but basically the most important thing you need to know is that this wasn't just a fluke, we found it again. So we had another study where participants perform actions that are probabilistically paired with grating orientations on the screen, and we found exactly the same. So superior decoding of those gratings that were expected on the basis of the preceding action relative to those gratings that are unexpected. I'm going to do an incredibly quick whistle-stop tour now of the fact that we've conducted a good 25 psychophysical experiments where we've taken the paradigms and analyses from outside of action to action. So these are paradigms and analyses that have resulted in support for these Bayesian accounts of perception, such that you're biased to perceive what you expect, so more likely to perceive it, and you're perceiving it more intensely. So basically across the board here in all of these studies, we've only ever then found support for these Bayesian upweighting type models in these types of paradigms where that Bayesian support has been found outside. So to give you a very quick idea of this, we had 3 signal detection studies where we present sensory events near the sensory threshold of participants and just ask them what they perceive. And we found biases towards perceiving what you expect on the basis of your actions. And then when we did some drift diffusion modeling, we found that this was due to biases in the evidence accumulation process. We did some contrast judgment studies where participants judged the contrast of the sensory action outcomes. And we found in quite a few studies here that expected visual action outcomes are perceived with higher contrast than unexpected. We had some studies looking at duration estimates, where you perform actions of different durations. So you reach towards close targets or further away targets such that you can manipulate the duration of your actions. And we played concurrent tones while they perform these tasks. And we found that when you're required to estimate the duration of these tones, that you're being biased in line with the duration of the action that you're performing. So if you're performing an action of a long duration, you think that this tone that you were played is of a longer duration than it actually was. And last, but certainly not least, there's been a lot of focus on touch in these action studies in the past. There are many plausible reasons why touch really might operate differently due to, well, very close relationships between motor and somatosensory cortex. This does not appear to be the case. So we found that actually when we use the same types of paradigm as have been employed outside of action, expected tactile action outcomes are perceived as more forceful than the unexpected outcomes. So when we've employed the paradigms and analyses demonstrating support for Bayesian accounts outside of action to action, we see similar support for these Bayesian accounts within action, which I think is demonstrating quite clearly from my point of view that theories concerning how predictions influence perception should not be opposite in the way that they are currently. And in fact, at the moment, we don't have any evidence that they should even be different. The next step here is, okay, so we've said probably these current models don't work so well. In fact, the models outside of action perhaps might not work too well either, because the models outside of action have been showing how you could render your experiences veridical in the face of noise, but are in fact proposing processes that in principle could be acting against the informativeness of our experiences. And in fact, it is crucial to organisms that if you present something highly surprising, they must dedicate a lot of perceptual processing resources to this event, because you need to update your models even if you're not moving. So if it's that you are in a certain sensory context and saw something that was inconsistent with that sensory context, maybe your models of how contexts go with objects is also incorrect. You need to update it. This is where we move into territory, though, where we're far more focused on possible solutions than something that we know about, because we have thought about it more than we have run the studies at this point. But so we published a couple of papers last year in Trends in Cognitive Sciences, where we proposed an in-principle solution to how we render perception both veridical and informative. I think this applies within action, but in fact, it is also a model that we think could apply outside of action as well. And what we've proposed is that we could extrapolate the solution perhaps from the learning and inference literature. So of course, learning researchers care about how we update our models of the world, but to do that, they also need to be considering a bit about perception at the same time. And of course, these are very intricately linked types of mechanism. So learning researchers have proposed processes that operate reactively to the presentation of a highly surprising event. And these proposed processes have been proposed to facilitate learning. So the types of process I'm talking here are phasic catecholamine release. So about 100 milliseconds after you present a highly surprising event, you'll have a phasic release of noradrenaline dopamine. Other studies demonstrating, and probably relatedly to this catecholamine release, saccades towards the unexpected, if you're free to move your eyes, also dilation of the pupils in response to presenting a surprising event. All of these types of process have been found to facilitate learning, but what we believe might be the case is that these processes are facilitating learning via an influence on perception. So they help learning by helping you to generate a better representation of these surprising events in order to help you update your model of the world accurately. So they may post-perceptually increase the sensory gain related to those very surprising events, such that you can increase the amount of sensory information you have about that event before you update your model. So you have enough sensory information to say something was different from what I thought, but now actually you can't just go with that. You want to increase the precision on that sensory representation before you update your model. And so our working toy model at the moment is that in line with these Bayesian accounts outside of action, we initially bias our perception towards what we expect to generate a broadly best guess accurate best guess representation of what is out there and rapidly. But if you present an unexpected input that is especially surprising, so hard to go into the details in the time frame, we think that you will need an especially high level of deviation between your prediction and what came in to elicit this process. But if you present something especially surprising, you have reactive processes that increase the sensory gain associated with this event, such that you can generate a better representation of it before you update your model. So we've spoken in the paper a lot about the Kullback-Leibler divergence, but so meaning that if your distribution between what you expected and what came in is pretty much overlapping, probably nothing much happens. But if these distributions are pretty much non-overlapping, And of course, if this is how it works, we need to find where that split comes. Then you will elicit this process to facilitate perception in order to facilitate learning. We haven't done much in the way of directly testing it yet, but we do have some data consistent with this idea, which is some experiments that Daniel ran during his PhD, whereby you execute actions and observe this avatar performing actions, and you rate the contrast intensity of these action outcomes at different time points after the action and the presentation of the stimulus. So 50 milliseconds later, 200 milliseconds later. And across 3 experiments, what we found is that 50 milliseconds after the action or after the sensory event, you perceive the expected outcomes more intensely than the unexpected, but by 200 milliseconds, this is flipped over. So these unexpected outcomes are perceived more intensely with higher contrast. So consistent with this broad idea at present. So to summarize, action predictions do not simply attenuate perception of outcomes. They're influencing perception via similar mechanisms to other predictions is our working model. We now need to test how we can make our perception of action outcomes veridical and informative, but we believe the solution probably won't just apply to action. And importantly, it's concerning this synergistic relationship between perception and action and how they have to serve each other. We need to test how this more nuanced picture of the actions determining perception is subsequently determining the action that follows. Before I thank everyone, just to note that we are recruiting too, like some of the other speakers. So this is very much the starting point for trying to figure out the solutions to this problem. So we got a 5-year grant starting in a couple of months now, modelling MEG and MRI. If you're a MSc or PhD student and are looking for a position across the next few years and like these questions, please get in touch. Thank you to everyone, so especially to the people who have conducted this work. So where Daniel and Emily conducted a very large portion of it, and then to my collaborators where Floris and Peter have collaborated on the majority of it too. So thank you very much.
Thanks a lot, Glenn. Any questions from the Raphael?
Thank you very much for the topic. So you presented the cancellation and the Bayesian counts as opposing.
Yes.
This is framework which can reconcile them, the predictive coding framework, according to which the brain, generates prediction and propagates prediction errors, but at the same time as it's only non-known, it can do Bayesian inference. So how do you try to think about your data in terms of prediction coding?
Absolutely. So one answer to that question is that this is a model of perception. So the predictive coding framework is a model of cortical processing, whereby you can have 2 units with opposing roles at all time points. You can only have one experience. So in fact, most people I know believe that the experience would be reflected in the hypothesis units rather than the error units. And therefore, the predictive coding framework in no way resolves this paradox, because as I say, you can only perceive one thing at a given time. You can have units with different roles, but this is part of the ERC grant too, is to ask how those units work together, and that's where the 70 MRI work comes in.
So I see that the way I would interpret data and drawing data is that when you have an unexpected event, you have a lot of error activity, but this value nodes which encode the activity are not encoding this information so precisely. Yes.
Yeah. But so the thing is, Carl has been very clear in papers that the percept is not the error activity. So I mean, certainly, yes, on these unexpected events, they should generate a lot of error activity. Of course, I mean, I'm on board in principle with the predictive coding framework, and but we don't even know at this stage that that's how the brain is working. So meaning that we are testing that orthogonally to this account, but so I would see this account as being orthogonal to it. So we are okay. It works with there being 2 unit types doing orthogonal things. But as I say, in terms of the perceptual experience for the organism, which is the level at which evolution needs to work, you only perceive one thing at a given time.
Yeah.
Thank you for the talk. I'm kind of wondering about these psychophysical experiments.
Yeah.
Comparing expected and unexpected events.
Mhm.
I would assume that cancellation, the evolutionary purpose of cancellation is not just to predict this kind of low-level model, but like even the unexpected event is a really unexpected.
Yes.
Is what I'm
Yeah.
saying. So how do you like? I mean, if suddenly there was a picture of an elephant, that would be
Yes.
unexpected. But there just seems a different grading, I think. Would that be a different system?
Absolutely. Yeah, no, I think no, in fact, this is a big part of what we have started doing, but it was not quite polished enough to present to you today. Absolutely. I think that the expectedness has been treated just as a sort of binary in many of these studies. Yeah, presenting a grading 25% of the time following a certain cue should not lead to any model updating. And so I know, so Peter Diane has focused on the difference between expected and unexpected uncertainty, which I agree with you is key here. So this is actually something that Kirsten, who has just asked for PhD with me, has been working on, is actually that some events should lead to learning and that should therefore require these sensory amplification processes, but look, most error is noise. So yeah, no, very interesting question.
Quick look on the chat.
Some questions in the chat.
So we have from Whaling Tan, whether a stimuli is surprising or not depends on the estimated uncertainty of the percepted information. How do you think the perception uncertainty is encoded in the neural activity?
Right, so trying to unpack that. So certainly I agree. So when we talk about surprise, so we've said in the ticks about being the KLD with respect to these two distributions. This is therefore not just the mean of these distributions, it's the precision on them. So it absolutely depends on the estimated uncertainty. How do you think it is encoded? So this is related to these big debates like the Susanna Siegel, Dobie Ranev, right, of whether the extent to which perception is probabilistic, perhaps. So we don't know at the moment. So certainly, with these pre-activation type stories and the lateral inhibition, it seems whether you can really activate this as all a very mottled landscape or whether it's a bit more binary than this and how that feeds through to perception. It was a very interesting question.
So we have one more. On the third last slide, besides sensory perception, an action you included language as an area worth investigating. How would you go about studying predictive language processing?
Yeah, very interesting question. So we are currently starting to think about these questions a little more, but so Matt Davis at Cambridge is one person whose work I know very well, whereby they look at predictive relationships between words that are read and words that are heard. And in fact, are looking at very similar types of models to us. So they present words that are consistent or inconsistent. So they present a visual word and it's followed by an auditory word that matches or doesn't match. And so you can predict at that level between modalities. In language, you can of course also predict within a sentence. So the statistical relationships within language means that when you hear certain words, other words are more or less likely to follow. So similar types of paradigm really can be used in language and are being used in language.
All right, thanks a lot. I think we'll call it to an end there. I hope you'll thank me in thanking Claire and all the other speakers.